Hello, good morning. Today we are going to discuss gene expression. And gene expression is exactly the process by which the instructions in our DNA are converted into a functional product, such as a protein. This gene expression is a part of the central dogma of molecular biology. As you know, the central dogma, which is actually proposed by Crick in 1958, it states that the DNA it is actually transcribed into RNA, and then RNA is translated into proteins. So that was the initial concept of the central dogma of molecular biology. And the information actually that is stored in the DNA in the language of the triplet codes is transcribed or copied into another language of the same or another language, or sorry, in the same language in the case of the transcription process. So the language is same. The language is the language of the nucleotides. So that's why it is known as a transcription. Whereas in the translation process, in which the mRNA is translated into the protein sequence, the language is different because the information is contained in the mRNA in the language of the nucleotides, whereas in case of the proteins that is coded from the mRNA, it is the language of the amino acids. So the letters were different. So that's why it is known as a translation. So the language is different. Okay. Now we know that the central dogma is revised later, incorporating various other aspects of molecular biology. You know that the DNA polymerase is the enzyme that is used for replication of the DNA. That is a production of a DNA copy from DNA itself. So that's the process of DNA replication. So the enzyme involved is a DNA polymerase. We will say that DNA polymerase is a DNA dependent DNA polymerase because the DNA dependent DNA polymerase depends on a template DNA. So using a template DNA, the DNA polymerase produces another copy of the DNA. So that process is known as DNA replication. Now when we come to the example of the transcription process, in which the RNA is produced from a DNA template, we use the enzyme RNA polymerase. So that RNA polymerase enzyme is nothing but it is a DNA dependent RNA polymerase. So DNA dependent means it depends on the DNA, or it uses a template DNA for the production of the RNA copy in case of transcription. So that's why normally the RNA polymerase, which is called in short, that is a DNA dependent RNA polymerase. Now we have the translation process, which is the third step in the or the next step in the central dogma. So in the translation process, the mRNA, which is normally having a positive sense, that is translated into a protein sequence by the ribosome or the translation assembly, and that RNA information is translated in the language of the proteins in case of translation. Later we have various additions in this initial central dogma, like the case of the reverse transcription process, which is later identified. In that case, we know that the mRNA or the RNA is actually used as a template for the production of a DNA copy of the genome. So that process is known as reverse transcription enzyme. That process is known as reverse transcription, and the enzyme is used is known as a reverse transcriptase enzyme. So that's now incorporated in the central dogma part. Now, there is another case in which we have a RNA replication process occurs, where the RNA copy is produced from the RNA template. So from the plus sense RNA, minus sense RNA is produced, and from the same, another copy of the plus sense RNA can be synthesized. So an RNA synthesis from an RNA template is known as a RNA replication, and RNA replicase enzymes are there, which actually copies the RNA from the RNA template, and they are RNA dependent RNA polymerases. And the previous case of the reverse transcriptase, they are RNA dependent DNA polymerases. Okay, so this is various aspects of the central dogma of molecular biology. Of course, there are some components like prions, which are infectious proteins, which is already reported and known. Mad cow disease is caused by prions, but they are actually proteinaceous substances, which we know that they are not information storehouses. Maybe it affects the structure of other proteins, or so that it becomes infectious, but it is not a storehouse of information like that of the DNA and RNA. So this is the overall idea about the central dogma of molecular biology. And now we know that already we discussed in the previous sessions the various structural components of the DNA and RNA, where we have the deoxyribose sugar and ribose sugar, that is the 5-carbon sugar that is used in case of nucleic acid. And we have a phosphate group. Then there are 5 different nitrogenous bases. They are coming under 2 categories, that is double ringed compounds, which is known as purines, and single ringed compounds, they are known as pyrimidines. And the basic difference between the RNA and DNA is that the thymine is used in case of DNA, whereas RNA uses a uracil instead of a thymine. And we already discussed the ATGC pairing and their GC content, all the ratios, etc. were already discussed in one of the previous sessions. So this is the structural components that is used for for the synthesis of DNA or RNA. So this is the phosphodiester backbone that is connecting the 5' phosphate and 3' OH of 2 nucleotides. So, and that bond is known as a phosphodiester bond, which actually creates the backbone of a DNA template. Now we have purines and pyrimidine bases, and you know that the pyrimidines are single ringed compounds, and uracil, cytosine, and thymine were single ringed compounds coming under the pyrimidine category. Purine is a double ringed compound, and adenine and guanine are double ringed compounds. And there are slight chemical differences are there, as we discussed in one of the previous sessions. Thymine is actually tagged with a CH3 tag here, whereas the cytosine, a deamination of the cytosine can result in the production of a uracil. That is also discussed in one of the previous sessions. So the difference between cytosine and uracil is nothing but a deaminated addition is occurring at this point. So instead of the NH2 group, here we have a O group here in the case of a uracil. So this is the structure of the pyrimidines and purine bases that is used in case of the DNA and RNA. Now when we come to the actual gene expression part, we have to discuss in detail the transcription process. As you know, the transcription is the DNA dependent synthesis of the RNA. And the process of synthesis of the RNA by copying the template strand of the DNA is known as a transcription. During the replication, the entire genome is copied, but in transcription only the selected portion of the gene or the genome or gene is copied. So the difference is that replication copies the entire genome, but the transcription copies only the required or selected genes which is present in the genome. The enzyme involved in transcription is a RNA polymerase, Unlike DNA polymerase, it can initiate transcription by itself. It does not require primase, and more exactly, it is a DNA dependent RNA polymerase. When we come to the different types of RNAs, we have a messenger RNAs, which is known as mRNAs, and mRNAs encode the amino acid sequence of one or more polypeptides specified by the gene or the set of genes that is present. So these mRNAs encode the sequence of the amino acids that is included or incorporated in the polypeptides that is synthesized. So the messenger RNAs actually carries the message for the polypeptide synthesis. Then transfer RNA or tRNA actually they read the information encoded in the mRNA and transfer that appropriate amino acid into the growing polypeptide chain during the protein synthesis. Then ribosomal RNAs are the third category, which is known as rRNAs, are constituents of the ribosomes and they integrate cellular machines, that is the ribosomes, that synthesize the proteins. So these are the protein synthesis machinery. Ribosomes are the protein synthesis machinery which is used for translation purpose, and these ribosomal RNAs are the structural components of the ribosomes that is used for translation. And to sum up, all the RNA molecules produced in a cell under a given set of conditions is known as a cellular transcriptome. So nowadays the terms like transcriptome analysis is more popular. For example, at a particular growth condition or at a particular environmental condition and a set of growth conditions, the organism will have a particular behavior which will be expressed, or that behavior can be explained by the different set of genes that is functional at that particular time. So the organism may have thousands of genes, but at a time, at a particular set of conditions, it will express a set of genes, and some of the genes may not be expressed at that environmental condition or that growth condition. So the set of genes that is expressed, or the mRNAs that the genes expressed means they produce mRNAs or they are translated and they produce proteins. So that particular set of the mRNAs that is coded or expressed at a particular time, they are collectively known as the transcriptome. Now we will discuss a little bit on prokaryotic transcription. Before moving with the prokaryotic transcription, we have to understand the sense and the antisense strands. Here, in this picture, we can see that the 5' to 3' strand is shown there, and the complementary strand which is arranged in the 3' to 5' direction, which is also shown here. And the first strand, this strand is known as a DNA non-template or coding strand or sense strand. And using this second strand, the RNA transcript is produced. So the RNA transcript, which can be direct, normally all the transcripts were marked from the 5' or 5' to 3'. So that is the normal representation of a nucleotide or a nucleotide strand. And here the 5' will be on the left side and the 3' will be on the right side. So in the left side, if you are arranging the RNA transcripts something like this, the template from which it is copied, the DNA template from which it is copied will be in the opposite direction, that is 3' to 5'. So that template, DNA template, which is used for copying this RNA transcript is known as a non-coding or antisense strand. So always keep it in mind that the non-coding or antisense strand is the actual template that is used for copying the RNA transcript. Non-coding or antisense strand is the actual template. So the other copy of the DNA, it is known as the non-template strand or coding and sense strand, because the RNA transcript is having the same directionality or the same sense as that of the DNA non-template strand, because it is having the same complementary sequence of the DNA template. So this DNA template is copied from this one, and this RNA is also copied from using this template. That means It is the DNA template strand is the non-coding or the antisense strand, and the DNA non-template strand is known as the coding or sense strand because it is having the same sense and directionality as that of the RNA transcript that is produced. Okay. Now, when we come to the transcription by the RNA polymerase in E. coli, we have a transcription bubble that is created here. Here it is the DNA, template DNA, and this is the template strand. This is the non-template or the sense strand, which is present here. This is the antisense strand or the template strand onto which the RNA is actually, or using this template strand, or the RNA transcript is actually produced. So this is the RNA polymerase enzyme that is actually unwinding that region, creating the transcription bubble. And here we have the RNA DNA hybrid that is produced. So this is the RNA DNA hybrid which is produced at the initiation of the transcription. So this is the direction of the transcription. Since the strand unwinding proceeds in this direction or from 3' to 5' end, then the unwinding will create a positive supercoiling on one end, and on the reverse side, the rewinding occurs when the RNA polymerase moves during the elongation stage. From the back end, there will be rewinding of the DNA template strand occurs, and that creates a negative supercoiling. Okay. So the transcription unit, it is defined as the stretch of the DNA sequence that is transcribed into a single RNA. So the region or the stretch of the DNA that is transcribed into a single RNA is known as a transcription unit. It consists of a promoter region at the beginning, and that is at the beginning means there is a 3' end of the antisense strand. Then there is a start point of a transcription, there is a coding region of the gene, and there is a termination sequence which is present at the 5' end of the antisense strand. So we have 4 different regions which is present in that transcription unit, or the region to be copied during the translation. So for the region that is copied during the transcription process. The first one is the promoter region. The second part is the start point. The third one is the coding region, and the final point is the terminator sequence, which is present near the 5' end of the antisense strand. And this is the representation of the transcription cycle in bacteria. Here we have the RNA polymerase which is binding to the DNA. So this is the DNA template, and RNA polymerase binds, and there is a chain initiation by incorporation of a nucleotide, any ATP or any nucleotide triphosphate that is incorporated as the first nucleotide of the chain. And here we have a open promoter complex. Initially, this is a closed promoter complex, and after the binding of the RNA polymerase and incorporation of the first few, maybe a few nucleotides, like 10 nucleotides initially, the closed promoter complex will create an open promoter complex, and then there is a promoter clearance. Promoter clearance means the factors, some of the factors like sigma factor will leave the promoter complex, and the promoter will be free to move, and that bubble, transcription bubble, can proceed in the direction of the elongation. So that is this is a promoter clearance is an next stage. So chain initiation by incorporating the initial nucleotides, then promoter clearance by incorporating the initial bases, and also by releasing the sigma factor and other things. Now then there is the elongation stage, chain elongation by incorporating the new nucleotides to the 3' end or 3' end of the growing RNA chain, and we have the chain elongation process. Finally, at the termination point, the chain terminates and the RNA polymerase is released, and the RNA is released, RNA polymerase is also released, which will be subsequently reused during the next cycle of transcription. So this is the overall picture of the transcription in the bacteria. You will see it in detail. So this is another representation in which the sigma factor will and the RNA polymerase, which will bind. So along with the sigma factor, the RNA polymerase core enzyme will bind to the promoter region, and it creates the initially it creates a closed promoter complex. Then it opens up the promoter, and also that region of the DNA helix, or the bubble, transcription bubble, will be opened, and a few bases will be added, initial bases will be added. Then it creates the open promoter complex, and the promoter clearance occurs, where the sigma factor leaves the system, and that release of the sigma factor actually it promotes movement of this RNA polymerase in the direction of the transcription. So that's what occurs here. So finally, you have a nacelle protein that is binding here, and once it reaches the termination sequence, at that point the completed RNA will be released at the termination point, and nacelle is also released. And finally, we have the recycling of these RNA polymerase for subsequent binding. So this is one of the representation of the RNA, the transcription process in case of a prokaryote. And the promoter sequence in a prokaryotic system, it is the start point, or the point from which the transcription begins. In 90% of the cases, the start point is a purine. Often it is an A residue in the triplet of CAT. In the triplet CAT, the A residue is there. In 90% of the cases, that purine, that A, is the start point of the transcription. Then there's another consensus sequences present. There are consensus sequences present at -10, which is known as Pribno box, and there is a -35 sequence in the case of prokaryotes, and especially in case of E. coli. These are AT-rich regions. These -10 and -35 sequences are AT-rich regions. -35 plays an important role in the promoter recognition by RNA polymerase. And the distance between the -10 and -35 is usually 10 to 18 base pair length. Here, it is an overall representation of the different regions in the transcription unit. So this is the transcription unit is the region of the DNA, or that is actually copied during the single transcription process, and that also includes is not the region that is corresponding to the coding region. There are non-coding regions at both ends, 5' and 3' end of the gene. That they are the regulatory elements. They are also part of the transcription unit. And this is the start point, that is the A in the CAT sequence, that is the +1 is the start point of transcription. And in the previous slide, we mentioned that is the purine, that is the A in the adenine in the sequence of a CAT. So this is the transcribed region. Transcribed region will be transcribed into the corresponding mRNA. And here we have the upstream sequences, or the sequences that is present upstream of the transcription unit. Here is the -10 region, and this is a -35 sequence. And the TA-AT is the -10 region. TG-TT GA-CA is the -35 region, and this is the coding strand. This is the template strand that is represented here, and from the template strand, the mRNA is actually copied, and starting from the start point, it is copied, and it ends at the termination signals. Then the flanking sequences, upstream flanking sequences, are shown here from this point onwards, or the sequences behind the +1 region is the 5' flanking sequences, and 5' untranslated regions were there, UTRs, and there are including the termination signals, there is a 3' flanking sequences, or 3' untranslated regions were also present in the transcription unit. So these are the general, this is the general structure of a transcription unit in a prokaryotic system. So the upstream and downstream, the terms should be identified. The upstream is towards the 5' end of the mRNA, whereas the downstream is towards the 3' end of the mRNA. So the term consensus sequences, what does it mean? The consensus sequences means these are the sequences that is reaching a common agreement. For example, if you analyze 10 different genes for their promoter regions, 10 different promoter regions from 10 different genes were simultaneously aligned and analyzed, you can see that there is some similarity in, for example, at the -10 position, there will be, for example, T. In 90% or 99% of the cases, T will be there at the -10 position. And at 99% or 95% of the cases, we'll have A at -11 position. And at -12 position, maybe we have another T. So if you compare a lot of sequences or promoter sequences from different species or different prokaryotic systems or different genes, and see what is the exact base that is presented at a particular position, -10 or -11 or -12, something like that. So that average or most predominantly occurring base at that particular position will be marked as the consensus sequence. So in prokaryotic system, the -10 promoter region will have a consensus sequence of a TA, TA, AT. So that is the most probable base that is expected to occur at that particular position, -10, -11, -8 or something like that. So in the case of -35, these are the consensus sequences that can be expected. In most probably, this will be the basis which is occurring at the corresponding position. So this is another representation in which consensus sequences were represented. This is the -10 region, and there are spacer sequences in between. And the spacer sequences, as previously mentioned, this may be 16 to 18 base pair difference will be there between these -35 and -10 region. So this N17 means 70 nucleotides were present in between these two, -35 region and -10 region. And here we have 16, and the N17 or N16 means we have 16 to 18 base pair spacer sequences were present between the -10 and -35 consensus sequences. Now we have some UP elements or UP elements which is present here in some cases, and this is the +1 point or the start point. And as we mentioned, in most of the cases, A or majority of the cases, the A in the CAT sequence will be the start point that is represented as the +1. And this is the example of a eukaryotic Tata box. The consensus sequence of that eukaryotic Tata box is represented there. TA, TA, AT, or TA. Then at the final position, AT or GC may alternatively occur. So that is the proportion of there may be varying proportions of the bases at some positions like AG or AT. Alternatively, both the items can be represented there. So this is the representation of the consensus sequences that is present in case of the eukaryotic data box. And here is the +1 site from which the transcription occurs. Now, the polymerase enzyme that is used in prokaryotic system, that is RNA polymerase, and that RNA polymerase is it is known as a hollow enzyme. The fully functional enzyme is the hollow enzyme that is having two alpha units, beta 2 or beta prime, then omega and sigma. So these are the different components of the hollow enzyme. So the core enzyme is the part which without the sigma unit, that is alpha 2, beta dash and omega. So this is the core enzyme, and the role of each unit is also mentioned there. the alpha is encoded by RPOA, and the assembly of the core enzyme is the role of that alpha. Two alpha units will be used in the core enzyme. Then beta is encoded by RPOB, that is it binds incoming nucleotides. Then beta dash encodes or it is encoded by the RPOC, and it binds to template DNA strand. Then omega, this is a omega, this is a capital letter omega, and omega encoded by the RPO omega subunit. That is a this is assembly, this assembly of the core enzyme is the role of this omega. Then we have the sigma factor, that is the final subunit which makes the holo enzyme, that is encoded by the RPOD, and this actually it stabilizes, or the stable binding to the promoter region of the DNA is actually supported by the sigma factor. So without the sigma factor, the core enzyme cannot specifically bind to the promoter region. So the specificity of the RNA polymerase holo enzyme to the promoter region or the +1 region or the region spanning the +1 region is ensured by, or that specificity is ensured by the sigma factor. So without the sigma factor, the RNA polymerase core enzyme cannot specifically bind and initiate the transcription process. So the sigma factor is a key component that makes the enzyme fully functional, and that sigma factor is required for specific binding at the +1 or initiation site. So in case of initiation, as we mentioned, the transcription is initiated at the promoter sites on the DNA template. The sigma subunit and the RNA polymerase core enzyme recognizes the promoter region. Without the sigma subunit, as I mentioned, the promoter region, especially specifically this promoter region or the +1 region cannot be specifically recognized by the core enzyme. The RNA polymerase unwinds the DNA about 20 base pair lengths. So that is the region of the transcription bubble. So about 20 base pairs of unwound region, unwound DNA is the transcription bubble. The RNA chains form de novo and grow in 5' to 3' direction. So de novo means it doesn't require any priming or some already existing free 3'-OH group is not required. Usually one initial nucleotide is actually used, and the 5' end of the or 5' phosphate of that initiator nucleotide will be untouched. Usually all the extensions or chain extensions or initiation in case of also DNA replication, for example, it starts with joining of a 5' to 3' or phosphodiester bond formation. Here it is not like that. The initial nucleotide that is coming in will have a free 5' phosphate group that is left untouched, and the subsequent nucleotides that is added will be added to the 3' OH of the first or initiator nucleotide, and we don't require any primer or primase enzyme or etc. is not required in case of transcription initiation. So the region of the initial unwinding by the RNA polymerase is about 20 base pair length, and that creates the transcription bubble. So here is a representation where the core holo enzyme is represented initially. That is the core enzyme plus the sigma subunit constitutes the core enzyme, which specifically binds to the initiator region or the starting point, and the binding of which creates a closed promoter complex that unwinds, and open promoter complex is created when about 20 base pair length of the DNA is unwound. So that is the after the binding, specific binding in the initiator region and formation of the closed complex, the strand is unwound, and that 20 base pair length unwinding of the DNA creates the open promoter complex. And in the next picture, we can see the stages of transcription or initiation is shown there. So that is the closed promoter complex here. This is the open promoter complex by strand unwinding and incorporating the first few nucleotides. So that is the next stage. First few nucleotides means about maybe 9 to 10 nucleotides were added initially by the RNA polymerase in this region. So that is the first initiation stage of the transcription. And usually after this initiation stage, maybe the transcription stops at that point without proceeding. So that type of termination of transcription without further extension or elongation can occur. And if it is not occurring or if it is not prematurely terminating, then the next step is the promoter clearance. So once the promoter is cleared by relieving or expelling the sigma subunit, the sigma subunit is the one that is actually giving a strong affinity for the RNA polymerase to the +1 region. So without the sigma subunit, the RNA polymerase is actually free to move from the +1 region or the initiator region. So that means the promoter clearance occurs by the release of the sigma factor. So now it states that it is not the exact, it is based on some structural changes in the DNA, not exactly by the release of the sigma subunit. So now there are difference of opinions on that one, but usually we consider that the release of the sigma subunit is a part of the promoter clearance, which allows the RNA polymerase to move ahead to the next stage, that is elongated stage. So this is how it occurs. So promoter clearance occurs, the initiation occurs, and after the synthesis of a short stretch of the RNA, then the sigma factor is released and the RNA polymerase moves ahead, elongating the RNA or the RNA transcript, and the sigma factor can again bind to the core enzyme and produce the holo enzyme, then start another process of initiation. So the next step is the elongation. The elongation is the second stage in the transcription. The extent of this transcription bubble, that is the DNA unwinding, is almost constant throughout the transcription and has been estimated to about 20 base pairs per polymerase molecule. Thus, it appears that the size of the unwound DNA region is dictated by the polymerase and is independent of the DNA sequence and its complex. That means the size of the size of the bubble or the length of the unwinding is actually determined by the polymerase. So the polymerase actually unwinds only about 20 base pair length region, so that actually constitutes the bubble. So that that length of unwinding is actually determined by the polymerase enzyme and not by the DNA sequence or the entire complex or the associated proteins. So this is the overall length of the transcription bubble. Now, when we come to the third stage, that is a termination process of the transcription, we have two mechanisms of termination that is reported. The first one is the Rho dependent termination, Rho independent termination or stem loop formation, and the second one is a Rho dependent termination, which is which uses a factor, Rho factor, a protein factor, for a termination process. So the RNA polymerase poses at a variety of DNA sequences. Usually RNA polymerase moves by transcribing the mRNA, but when it encounters, for example, a GC-rich region, it slows down. If it is translating in an AT-rich region, the strand unwinding will be much easy and it will be much faster. So RNA polymerases usually poses or it slows down at a variety of DNA sequences. Two possible outcomes will be there. So first possible outcome of this transcription process is that the polymerase bypasses the site or these objections or obstructions which is faced by the RNA polymerase. It bypasses the site and continues on its way, or the complex undergoes a conformational change or isomerization and then it falls off. So we have two possibilities. If the RNA polymerase encounters an obstacle or a hurdle in the process of transcription, and we can expect two possible outcomes. One is bypassing that outcome. It overcomes that hurdle. Then it starts or it goes on with the elongation process. Or such hurdles may result in, or such sequence hurdles or the blocks will result in a conformational change of the newly synthesized RNA, and that isomerization process will result in termination of the transcription process. In the later case, that is the conformational change case, here we have the first stage. For example, this is the transcription bubble and this is the newly synthesized mRNA which is shown there, and usually it overcomes the hurdles and it bypasses or it goes on transcribing the RNA. And if there is a structural change like the formation of a stem loop, then it proceeds to termination instead of going for the or is going along with it, the elongation stage. So that's what happens. Okay, we can see that in the later case, intramolecular pairing of the complementary sequences in the newly formed RNA transcript may form a hairpin that disrupts the RNA-DNA hybrid and or the interactions between the RNA and the polymerase, resulting in the isomerization. So it actually forms a hairpin loop between the sequence of the RNA itself. So the newly synthesized RNA will have some self-complementary regions, and that regions will pair. And here in the case of the RNA transcript that is shown here, you can see that the RNA pair forms a base pairing between its own strands, or self-complementary regions of the RNA is paired together, and there is a loop region where pairing is not there. So the structural change that is caused by such a loop formation in the RNA will result in the loss of RNA DNA hybrid regions. Usually the newly synthesized RNA is kept in its position or in the replication bubble, transcription bubble by RNA DNA hybrid pairing. But when such loops or some other isomerized structures forms, the RNA can no longer pair with the complementary sequence in the DNA. So the RNA DNA hybrid will not be there, leaving only a very few bases to be attached with the DNA template. So and usually that regions will be some AT-rich regions. So a few bases cannot hold this long or growing RNA chain to the the transcription bubble. So that means it will fall off. So the structural change that is caused by the isomerization process or the stem loop formation results in the loss of the RNA DNA hybrid pairing in the transcription bubble, and the result is that this RNA, newly synthesized RNA, is no longer able to stick onto the DNA template in the transcription bubble. So that means it falls off. An AUU hybrid region at the 3' end of the new transcript is relatively unstable. So usually at the end there will be AUU, that is A and U residues were weaker bonds. So that regions which is holding these transcript to the DNA, that sequence AAU sequence were relatively unstable because it is AT-rich, so or AU-rich, so that they are weak. So the RNA will fall off from the hybrid template, and this is the usual outcome at the terminators. At other post sites, the complex may escape after the isomerization step to continue the RNA synthesis. If such an isomerization and falling off or the termination of the process is not occurring, and in that case, the RNA polymerase bypasses that region and it escapes that termination point, and the RNA synthesis continues. It is not terminated there. Now, the second category of termination is a Rho independent termination. Sorry, this is the same one. This is the same picture. Here we have the self-complementary regions which is represented here and here. The self-complementary regions will pair together, and the GC-rich regions is the self-complementary regions which will pair. And there is another AT-rich region here, which is shown here. So when this region is coded, the GC-rich regions will pair together to form the stem of the loop. And the in between N and that residues, that region is actually forming the loop part. And finally, the AT-rich region, which will code for multiple U residues in the stem loop. And the result is that the multiple U after the formation of the stem loop, the multiple U residues or the weak U residues cannot stick onto the growing or the transcription bubble, because it is only a weak bond which is unable to keep the long RNA transcript to the transcription bubble. So that means that after the formation of the stem loop, this structure will fall off. So this is the Rho dependent termination. That is the this is the same thing. The previous it's another representation of the same. This is a stem loop, and this is the multiple U residues which is holding to the DNA RNA hybrid. And this bond will not be sufficient for holding this RNA transcript to this bubble, so it falls off. Okay. So the second category is a Rho dependent termination. And Rho dependent termination involves a Rho factor which is an ATP dependent helicase. And it catalyzes the unwinding of the RNA DNA hybrid. So there's Rho factor, which is a ATP dependent helicase. Actually, it unwinds. The role of this helicase is unwinding of the RNA DNA hybrid. And it binds to C-rich regions of the RNA transcript, and it moves along the RNA chain to the transcription site or the bubble. So the transcription or the termination requires a specific sequence on the nascent RNA upstream of the termination site. So usually it requires a specific sequence, and we will come to that one. So anyway, this Rho factor is an ATP dependent helicase that is having the ability to unwind the RNA DNA hybrid that is formed during the transcription process. And it usually proceeds or it follows the RNA polymerase. You can see that the RNA polymerase actually moves along the DNA template, and this is the direction of growth of the bubble. And the Rho factor, which will bind to the growing RNA chain, and it proceeds or it follows the RNA polymerase in a very faster manner. Usually the rate of the Rho factor is much higher. And when the RNA polymerase encounters some GC-rich regions or some specific sequences which halt or which slows down the RNA polymerase slows down, and the following or preceding Rho factor catches up with the RNA polymerase, and that once the RNA polymerase is caught by the preceding or following these Rho factor, the RNA DNA hybrid is actually unwound or the pairing is lost, and then this unit will be relieved from the bubble, and the newly synthesized RNA is relieved, and the RNA polymerase and the Rho factor, all these things will be released, and this will be available for reuse. The Rho dependent terminators lack the sequence of repeated A residues in the template strand, but usually it includes a CA rich sequence called Rho utilization element, RUT. So this is one of the important factors where it doesn't have an A rich residue, but there is a CA rich sequence which is known as Rho utilization element. A specific sequence is there. When it encounters these RNA polymerase reaches at that point, it slows down, and this GC rich region slows down the RNA polymerase, allowing the Rho factor to catch up with the or catch the RNA polymerase, and that once it reaches the RNA polymerase, the entire DNA RNA hybrid will be released released from the bubble, and that is the Rho dependent termination process. So this is all about prokaryotic transcription, and in the next session we will discuss eukaryotic transcription and its regulation. Right now, we can stop for the time being.